Good morning, I'm Andrea Corinza and I'm going to present my research about soil water availability on biomass yield and water use efficiency of perennial grasses in semi-arid area. The research has been carried out in Sicily, in an area that falls within a classification of marginal lands that are affected by low soil water availability during summer. Experimental design is a split plot design with free replication. The main factor is irrigation with 3 levels that are 100%, 50%, and 0% of maximum crop evapotranspiration restoration during the summer month, in which the soil water availability is at the lowest level. The subfactor is a genotype, and we tested 6 genotypes. 2 ecotypes of Arundodonax that are named ARCT and ARMO, the Miscanthus pergiganteus hybrid, 2 new seed-based Miscanthus hybrids, and Saccharum spontaneum. The trial has not been fertilized, and we carry out 3 harvests, and I'm going to present the results for the harvest of the second growing season in 2019 and third growing season after establishment in 2020. Arundodonax is a seed-free perennial rhizomatous grass that is naturalized in the Mediterranean basin, which spreads only by vegetative propagation. We test 2 ecotypes, ARCT, which is a local ecotype, and ARMO, which has been collected in Morocco. We had 3 genotypes of Miscanthus pergiganteum. We test the Miscanthus pergiganteum hybrid and 2 new seed-based hybrids that have been developed by Ibers and Terravesta. These 2 hybrids comes from a parental selection for drought-tolerant traits. Saccharum spontaneum is a seed-free perennial rhizomatous grass that is naturalized in Sicily. The maximum crop evapotranspiration has been calculated using the potential evapotranspiration calculated by the Penman-Montier method and a crop coefficient based on the estimation of leaf area index. Irrigation volume has been calculated in order to provide the 65% of field capacity and accounting for a rooting depth that was 1 meter for Arundodonax and Saccharum spontaneum and 60 cm for the Miscanthus genotypes. During 2019, rainfall was above the average for the area, especially during the growing season, while the potential evapotranspiration has been close to the average. The same for the average temperature for the whole year and the growing season. The irrigation provided was 250 millimeters for the I-50 thesis and 490 millimeters for the I-100 thesis. In 2020, instead, the rainfall was slightly above the average for the area, even during the growing season, with high precipitation in March and September, while the potential evapotranspiration and the average temperature was close to the average. The irrigation was slightly more than in 2019. This plot represents the total above ground biomass yield. Here we can see that for Arundodonax genotypes, the yield were higher than for the other genotypes, and there was a strong response to the irrigation level. Only Saccharum spontaneum had a biomass yield higher for the I-50 thesis than for the I-100 thesis. The biomass yield for Saccharum spontaneum in the I-50 thesis was higher than the I-100 thesis, perhaps due to logging that occur at the highest irrigation level. Here we present the crop water use that has been calculated as the difference between the soil water content at the shoot emission and at leaf senescence, with the addition of rain and irrigation. Leaf senescence occurred in October for the Miscanthus hybrid, while in Arundodonax and Saccharum spontaneum it occurred 1 month later. In 2020, crop water use was higher due to the increase in rainfall and the slight increase in the irrigation amount that has been provided. Arundo and Saccharum spontaneum have a slightly higher water intake due to the deeper root system. This plot represents the crop water use efficiency that has been calculated as the ratio of biomass yield and crop water use. Higher values of crop water use efficiency has been observed for lower irrigation amounts. The only exception are in ARMO 100% in 2019 and in Saccharum spontaneum 50% in 2019. That results from high yield and a low water input that came from lower rainfall during that year. Water footprint gives an estimation of the amount of water that is necessary to obtain 1 ton of dry biomass. It has been calculated as the ratio of crop water use and the biomass yield. The lower footprint are observed for high-yielding genotypes and at the lower water input. The highest footprint is observed for GNT10 in both 2019 and 2020 at the 50% of water restitution because of the low biomass yield obtained. This research showed that Arundodonax and Saccharum spontaneum are better suited for Mediterranean marginal area that are affected by summer drought, both in irrigated and rain-fed condition. For all the genotypes, irrigation triggered a strong response in all the productive traits and in the water use efficiency. Arundodonax in both genotypes had the best biomass yield production and the highest crop water use efficiency, despite the C3 metabolism. This is due to the higher leaf index and a longer green leaf direction. The high performances of Saccharum spontaneum comes from the C4 metabolism and also from the long green leaf direction. Thanks for your attention.